Assalamualaikum warahmatullahi wabarakatuh and hi everyone. My name is Nur Azlida binti Muhammad Aziz from Sekolah Menengah Sains Raja Tun Azlan Shah. So in this video, I will explain to all of you the last subtopic for KSSM Biology Form 5, Genetic Technology, which is the last subtopic 28.2 Biotechnology. Before we start, let's take a look into the learning standard. There are 3 learning standards for this last subtopic, which is students should be able to explain the meaning of biotechnology. Second, students can be able to describe application of biotechnology in life. And lastly, students can justify the importance of biotechnology in life. So, what is the definition of biotechnology? It is a field of biology which utilizes technology or method to manipulate organisms for the production of biological products. The aims of biotechnology is to improve the quality of livestock and crops. So, if we produce a new quality of crops, so it is a biotechnology. Other than that, biotechnology also aims to develop the application of microorganisms for specific purpose. Biotechnology also using a living organism for specific purpose. Next, the application of biotechnology in life. Actually, there is a lot of application of biotechnology in life. But in this syllabus, we will take a look on the 4 application of biotechnology in life. So I hope all of you can explain each of the application of biotechnology. Okay, the first application is gene therapy. Gene therapy used to treat genetic disease caused by mutation in a single gene. A treatment of a genetic disease by changing the genes in a person's cells. Okay, what is the example of genetic disease? There are a lot of genetic disease. Some of them is such as sickle cell anemia, cystic fibrosis, Huntington disease. Okay, so this disease can be treated by using gene therapy. Okay, for example, we take a look for sickle cell anemia. You still remember what is sickle cell anemia? So it's one of the disease which caused by a gene when a person has an abnormal gene and produce a red blood cell which has a sickle cell shape. Okay, so how to treat this disease using gene therapy? First of all, we need to get the cell of the patient cell that contain with an abnormal gene. Okay, if you can see here, a cell with an abnormal gene which is only can produce sickle cell shape of red blood cell. Then, using gene therapy, we'll insert a normal gene to replace the abnormal gene using a virus. So, we'll insert a normal gene that contain the genetic to produce a biconcave shape of red blood cell into the patient. And as the result, the cell of the patient will perform a normal function and produce a red blood cell which has a biconcave shape, not a sickle cell shape. Okay, so this is the application of gene therapy. Next is DNA profiling. So DNA profiling is a technique used in forensic to identify individuals based on their DNA. This is because a human DNA set is different from animals and all human has a different set of DNA from each other. So what is the using of DNA profiling? Is for identify criminal, settle paternity and maternity dispute, identify genetic disease, determine suitability of organ donor and recipient. So how this step used in DNA profiling? First, we need to get a sample from a person that we want to investigate. So the sample can be a blood, semen, a skin, a hair, a saliva, or anything from the person. Then, from the sample, we will extract the DNA. So, some of the steps we'll use to get a pure DNA from the sample. After that, restriction enzyme will cut the DNA into different sizes of DNA fragments. So from the pure DNA sample, restriction enzyme is used to cut the DNA fragment. After the DNA fragment of different size get, so it will then separate by jet electrophoresis. So electrophoresis will separate the DNA fragment into same size. After that, the DNA fragment of different size are transferred from the gel to a nylon membrane for better seeing, and radioactive probes are added to the nylon membrane. X-ray film is then placed on top of the nylon membrane. And lastly, X-ray film is processed to show the position of DNA bands that form the DNA profile. So here is the DNA profile get from the sample. So from the DNA profile get, the forensic will compare with the suspected person. The third application of biotechnology in life is production of pest or herbicide-tolerant plant. This means we produce new plant that is tolerant to pest or herbicide. There are a lot of examples of GM crops. Example is GM cotton, GM soybean, and GM corn. So here in this table is the comparison between normal crop and GMO crop. Okay, this is the normal crop before we'll change it into GMO crop. So, BT cotton is the new plant that will produce which is tolerant to the pest of herbicide. Farming cotton breed with high yield and resistant to pests such as BT cotton in cotton farm has reduced the use of pesticide. The BT crop is injected with genes from Bacillus thuringiensis, BT. So that's why the name is BT cotton. So Bacillus thuringiensis is a type of soil bacteria that secretes toxin. So these genes had been injected with the gene of the cotton. So this effort has saved costs by reducing the use of pesticide and also increase cotton yield. So the production of new BT cotton help us to produce more quality cotton, and also a plant with have safe costs which reduce the use of pesticide. The last application of biotechnology in life is cleaning of oil spill. This technology using the microorganism. Cleaning of oil spill is a bioremediation method that uses bacteria to clean up environmental pollutant. It is involved in controlling environmental pollution through biodegradation of potential pollutant or recycle of waste. Most molecules include oil and purified oil products can be degraded by bacteria. The bacteria may produce specific enzyme and undergo certain chemical process that can break down contaminants. For example, the use of bacteria alkanivoride borkumensis, a type of bacteria that depends on oil for its source of energy. So, this bacteria is widely used to treat oil spill. So, we have finished with the application of biotechnology. Lastly, we'll take a look into the importance of biotechnology in life. There are a lot of contribution of biotechnology to life. For example, in the field of agriculture, biotechnology produces the best livestock and crop breeds. The tissue culture method and cloning technique have expedited plant reproduction. So, biotechnology ensure we only produce paddy varieties which give more yield and mature faster. Animal cloning enable mass reproduction and produce disease-resistant clones. Use of hormone expedite animal growth and maturity. Other than that, in the field of medicine, Biotechnology can modify the genetic contents of an individual, so reduce risks of mental disease. The application is, for example, like gene therapy. So it prevents specific disease such as cancer and inherited disease. Human with rough genetics can have normal children, assist mother with difficulty to conceive. Third, in the field of forensic, DNA profiling can identify criminals based on tissue or body fluids such as blood, semen, skin, or hair follicle. And lastly, environment. In the field of environment, biotechnology helps to use microbes and can expedite decay of waste product created by humans. Microbes can also degrade feces completely and reduce pollution to protect humans and the environment. So that's all for me. Thank you.